So the question is, how does the ribosome catalyze peptide bond formation? Okay, so is it chemical catalysis? So chemical catalysis is basically how every enzyme we know about works. And so what do I mean by chemical catalysis? It means the formation of bonds. They can be covalent bonds or they can be non-covalent bonds between the enzyme, between the catalyst and the transition state that stabilize the transition state. So we talked about how, for example, RNA polymerase and DNA polymerase function as metalloenzymes. They have metal ions that bind to the transition state and stabilize the transition state. Okay, so we form these coordinate bonds in that case, but in general, enzymes can make many different kinds of bonds with the transition state. The point is that these bonds always stabilize the transition state relative to the ground state. One thing that I'll be coming back to that you should remember is that bond formation, whether we're talking about ionic bonds or covalent bonds, or even dipole interactions, always results in a release of heat into the environment. In other words, bond formation is exothermic. Okay, and so this is another way of saying that bond formation decreases the enthalpy of the system. Because you might have learned in freshman chemistry at some point that the enthalpy change is just the change in the heat of the system at constant pressure. So as long as you're at constant pressure, the change in enthalpy is the same as the change in the heat energy, right? Okay, so here is a look at what's happening during peptide bond formation. We've already looked at it before. We have an aminoacyl tRNA and a peptidyl tRNA, and we form a tetrahedral intermediate. And here's the transition state on the way to the tetrahedral intermediate. So we need to, if we're going to think about chemical catalysis, we need to think about how we can form bonds to this transition state that would lower the enthalpy of that transition state relative to the ground state. So one idea that people had when they first started thinking about how catalysis might occur is the idea of general base catalysis. So the idea is that maybe there's a basic RNA residue in the active site, in the peptidyl transferase active site, that helps to pull this proton off of the nitrogen here, off of the amino group here, activating that amino group for a nucleophilic attack, right? And so, in other words, in the transition state, then we'll be forming a covalent bond between this base and the transition state, and thereby stabilizing the transition state relative to the ground state. Okay, but the problem with that is that when we look in the active site, we don't really see any residues that are properly positioned to act as general base catalysts. The only exception is adenosine 2486 of 23S ribosomal RNA, which is almost positioned correctly to serve this role. But the problem is when they mutated A2486, there was no change in the catalytic properties of the ribosome. Okay? So that seems to rule out the idea of a general base catalysis. Another possibility is that the ribosome uses metal ions to stabilize the transition state, very similar to what we've observed for DNA polymerase and RNA polymerase and the spliceosome, right? And so the idea is that there might be metal ions in the active site that coordinate atoms in the transition state and thereby stabilize the transition state. But the problem with that is that we don't see any metal ions in the active site when we look at the high-resolution crystal structure of the peptidyl transferase active site. Okay, so in the case of the ribosome, then, there's no clear evidence for chemical catalysis. And in fact, the data on the next slide that I'll go through with you in just a second actually seem to prove that chemical catalysis is not occurring. Okay? And how do we prove that chemical catalysis is not occurring? Well, if chemical catalysis is occurring, then the catalyst should lower the enthalpy of activation by reducing the enthalpy of the transition state relative to the ground state. And so we can measure the enthalpy of activation for the uncatalyzed reaction and compare that to the enthalpy of activation for the catalyzed reaction to see if we see a reduction in the enthalpy of activation in the presence of the catalyst.